So the other thing about our codons is that there's actually many more codons than there are amino acids. So because there's lots of different ways we can have these three base sequences. For example, we can have UAT, UAG, UAA. There's so many differences because we can have differences in all of these three positions. That means there's actually many more of these codons or codes than there are amino acids, which actually means that more than one code will lead to the same amino acid being added to our protein chain. So, for example, we've got two codes here. They both might lead to making alanine, the alanine amino acid. So that's why we describe our code as degenerate. More than one code leads to the same outcome, same amino acid. If every single code led to a different amino acid, then the code would be specific. That is not the case, so that's why we call it degenerate. Alright, so this is where we actually see how we use these codes. So the codon is that sequence of three bases within our mRNA. So within our mRNA, there's going to be heaps of these different codons or three-letter codes, and they're read by the tRNAs and the ribosome one at a time. So what we have is we have the codon from the mRNA, and then we have the anticodon from the tRNA matching up. Because the bases are actually going to be complementary, we actually get the formation of hydrogen bonds between each of these bases. So that's how the code on the codon, the code from the mRNA, actually dictates which amino acid is going to be added in. Because that particular codon in the mRNA actually specifies which tRNA comes in. So the tRNA contains a specific amino acid, and that's the amino acid that will be added in. So we can see the amino acid up top here. We've got the codon from the mRNA matching the anticodon, so that particular amino acid is going to be added to our chain. So we've got this being amino acid 1. So the next codon that's read, for example, if we've got the next one in the sequence, this might be it might be UUA. Okay? So this actually would bring in another tRNA, and that tRNA would have another amino acid. So this time we're adding in that amino acid, and it's going to be amino acid number 2. So we link these two together, and then we go to the next codon. So you can see how by going down the mRNA chain and having our tRNAs matching up with those, that is going to allow us to create a long chain of amino acids. And a long chain of amino acids is what we call a protein. So the sequence in our mRNA tells us which tRNAs to go and get, and they bring in specific amino acids, which is going to determine what ultimately the protein is that is made. So we remember we do this translation sequence from the start codon to the stop codon, and that's how we know where to stop the amino acids being added on. Remember that this is all happening at the ribosome. So the ribosome is the site of protein synthesis. So there's a nice summary here showing us how our original DNA actually ends up telling us which protein is going to be made. So we start off with the DNA, which has a particular sequence. We then copy that sequence to the mRNA through our process of transcription. And then we take that mRNA out into the cytosol, where we can match up with the ribosome and also the tRNAs to convert our mRNA code into our specific protein. And remember, the code on the mRNA tells us which amino acids to actually add to the protein. So we can have problems with our DNA or errors in our DNA structure. This can happen during the process of DNA replication. So basically, what we define as a mutation is an error in the base sequence. And this has the effect of changing the structure of the DNA. So for example, if we have a particular DNA sequence that is AATGCC, so this is the normal sequence. But what happens if in one situation, instead of getting the AATGGC, this is actually now changed to ATAGCC. So that A is changed, T is changed to an A, and that is actually what we call a mutation. So the mutation has caused one base pair to change, one base to change to another base. So this is what we call a mutation, and this changes the sequence of the DNA. So because we are actually changing the sequence of the DNA, basically what you're doing is you're changing the instructions that will ultimately lead us to get to the protein. So by changing the DNA, you change the mRNA that sequence as well, and ultimately you change what amino acid is actually going to be added on where we had that mutation. So this can actually potentially lead to a change in the function of the protein, and depending on what that protein is, it might be extremely important to your survival. This could actually be lethal. So this is why some fetuses actually never reach a certain stage of development. If they have one of these mutations, then basically they just stop growing and die. So there are certain genes and proteins that we actually have to have being functional, otherwise we just cannot survive. And these mutations are also the seen in a lot of hereditary diseases. So things like phenylketonuria, cystic fibrosis, sickle cell anemia. There's actually an error in our DNA, or the people who have these conditions, which means that the proteins that are encoded by that particular gene that there's a problem with actually don't function as well as they should, which means they have the condition or genetic disease which results. So basically we can summarize this by having altered DNA that causes the altered protein, and the consequence of that is sometimes we get the genetic disease because the protein is not working as well as it should, and that can lead to some problems. For example, in sickle cell anemia, the red blood cells don't form as well as they should, and they're not as good at carrying oxygen. So that's a problem for these individuals. So I'm sure you've heard a lot about cancer, and we can actually have genetic predispositions to cancer, but you're not born with cancer. Basically, what cancer is where a cell loses control of its replication processes. So normally there are tight regulations about when a cell will and won't divide. But what happens in cancer cells is we actually have mutations in specific genes that actually mean it's going to just keep replicating itself and not be controlled. So you can think of cancer as uncontrolled cell growth. So that's why we get these large tumours forming once we have cancer, because we've lost control of the growth of these cells. So there are a couple of types of mutations that I want to talk you through. So we've got the substitution mutations and also the frame shift mutations. So the substitution mutation is just a single change in the amino acid sequence. So similar to what I showed you on this previous slide where we have the ATTGCC, if we instead change that T to an A, this is what we would call a substitution mutation. You're subbing one amino acid, one nucleotide for another nucleotide, and this will ultimately be reflected in the protein that is made from this piece of DNA, because that will likely change the amino acid that is ultimately going to be added to your protein chain based on that codon where the mutation was a part of. So these mutations sometimes actually aren't very harmful because it's just one single alteration. So quite often this is not harmful. However, if we get what we call a frame shift mutation, this actually is a lot more problematic. So what happens here is, instead of changing one of our bases, say from an adenine to a thymine, what we actually have is we have a particular nucleotide either being lost or added in. So instead of having our AATGGC, let's say we now add in another one. So instead of being AATTGCC, we have AATTGCC. So we've added this extra nucleotide. So that means that instead of reading the codons as AATGCC, the codons are actually now read as AATTGC. So you can see it completely changes the order with how you're actually going to read your codons. So every codon downstream of the addition or subtraction of the base will actually be changed. So in our single mutation, we actually only change one codon, but when we're doing a frame shift, we change every single codon after the addition or removal of that base. So these are much more drastic and, in general, much more problematic. And it's going to lead to more than likely, it's going to lead to a non-functional protein. So what can actually cause these mutations? So mutations can be caused by what we call mutagens. So these can be chemicals or other agents which mutate the genetic material, which is our DNA. So things like ionizing radiation, particular chemicals like nitrous acid. We also have carcinogens. So these are our mutagens which are specific to mutating genes that are involved in cancer. So for example, the body has mechanisms to repair DNA, so not all mutations are going to be passed on to the next generation of cells. So if we get mutations in these repair enzymes, that actually is one of the things that can lead to the uncontrolled growth of the cells being cancer. So if we're not able to repair mutations, then this can actually lead to cancer. So the problem with this is that those mutations that are occurring are going to be able to be passed on to the next generation. And once we get lots and lots of these mutations, then the cells lose control of their replication and we're going to have this uncontrolled cell growth. So some other things that can cause mutations, things like viruses, which can actually be incorporated into the genetic makeup of the cell, and that can actually cause mutations as well. Other things like asbestos, which used to be used in building, and pesticides, smoking and alcohol also. So lots of different things can cause these mutations. But to cause cancer, the key is that we actually need lots of mutations in specific genes that control the cell cycle. And it's not just one mutation that's going to instantly cause cancer. We need many, many of these mutations. So this is one reason why the development of cancer actually takes such a long time, because we need lots of different mutations to actually lead to the uncontrolled cell growth that we see in cancer. Alright, so the last thing we're going to talk briefly about is the use of genetic engineering and recombinant DNA. So we call a recombinant piece of DNA basically the genetic material from two different organisms. So if we have the DNA from a monkey and we combine that or attach it to the DNA of a horse, then what we've actually created is recombinant DNA. We've fused bits of DNA from two different species together. So sometimes scientists will do this to try and create a new gene that has a particular function or that will be particularly favorable for a particular process. So scientists have actually used these characteristics to mix together the DNA of different plants to give them desirable consequences, whether that be increase in yields from harvesting, better resistance to drought, these types of things. So we've also used genetic engineering for other things like creating insulin from bacteria, which is very useful for diabetics. So another technique that's related to genetic engineering is called polymerase chain reaction. So this technique basically takes a very small piece of DNA and can actually replicate it millions of times. So we can actually use this to try and identify a particular pathogen. So all of our pathogens have some sort of genetic material associated with them. So by categorizing the different DNA sequences for each of these pathogens, we can actually work out what someone is infected by from taking a sample that includes cells from this pathogen, or if it's a virus, just the virus particle. So the way this polymerase chain reaction actually works is we start off with one helix of DNA. So the helix of DNA contains these two different strands. We first split it apart and then start to create new strands. So just like we did in our DNA replication, we're using the parent strands as new templates to make new strands. However, the difference here is we go through lots of cycles where we break apart the old strands and create new strands. So in this way, we're able to get many new strands of that DNA. So the polymerase chain reaction is just a way of creating lots and lots of copies of DNA from a very small amount of DNA. And we can actually do this very quickly. So within an hour or two, you can actually have created millions of copies of your DNA. So that gives you a lot more of your DNA to do tests with and to categorize. So it's a good way to make use of the DNA that you've harvested from a particular location. You can make lots of copies of it. So if you need a sample of it in the future, you can have access to that. So the other thing I want to show you here is to do with our recombinant DNA technology. So we have what we call plasmids, which we can actually insert a particular gene into. So the plasmids exist in bacteria like E. coli. So by putting in a particular gene into this plasmid, we can actually put it back into the E. coli, and then the E. coli will actually make lots of copies of that plasmid, and then it will start using the DNA in that plasmid to make proteins. So we can actually use this method to actually make lots of a particular protein that we're interested in or we need for a particular commercial purpose. For example, this is actually how we were able to produce insulin in for diabetics. So by putting the insulin gene into our plasmid and then putting it back into the E. coli actually make the insulin protein, which then we can take out through specific procedures. So we do a purification procedure to remove the insulin and get rid of everything else. So when would you want to do your polymerase chain reaction? We talked about trying to identify a particular pathogen in the body. If there's a particular genetic disorder that the individual may have, then we can do the polymerase chain reaction to generate a large amount of the DNA from a small sample and then do the different tests on that. We can also use it for forensics, trying to identify an individual from DNA that's left behind the crime scene. So here we can see some of the different disorders which we've actually been able to treat using this recombinant DNA technology. So we talked about diabetes, but also things like cancers and heart attacks. So while these recombinant DNA technology treatments are useful, they often aren't a complete solution. So we haven't cured cancer through the use of these, but it can actually help to slow the progression of it. Alright, so just to review some of the things we've looked at today, we firstly looked at our nucleic acids, so there were these two different types, our DNA and our RNA, and it was our nucleotides that actually make up each unit of our DNA and RNA. So within each nucleotide we have a phosphate, a sugar, which is a ribose or a deoxyribose, and then we have that nitrogen containing base we've talked a lot about, which forms these complementary relationships through hydrogen bonding. So this is the basis of our DNA double helix, which are held together through the hydrogen bonding between these nitrogen bases from the different strands of the DNA. So we've got two strands that come together and wind around each other, and they're held together through these hydrogen bonds between the nitrogen bases. We then talked about our DNA replication, which is our method of copying our genetic material and passing it on to the daughter cells from the parent. And then we had a look at our mRNA, rRNA, and tRNA, which we used to take that instruction from the DNA and actually create a specific protein. So we talked about the hypothetical example of the make more glucose protein, and the instruction for that came from the DNA, and we passed that all the way onto the protein, which was the make more glucose protein, which had the ultimate outcome of making more glucose for the cell. So I'll see you next time for our protein lecture.